Okay. Getting set up to start the live stream. I'm going to wait a few moments for people to tune in. Oops. Okay, I see some people dropping in. Great. Hello, everybody. I hope you've been having a great holiday weekend. Get ready for our last week of school here. Nice to see everybody. Hello, hello, hello. Okay. Let's see, before we get started, I just want to give a big old shout out to my mom. It's her birthday today. So if you're watching this, Mom, or watching it later, hope you're having a very happy birthday. I love you very much. Thank you for everything you're always doing for me to make it easier for me to do this sort of thing. Okay, so let's see. Whew, final exam. Everybody getting ready? Thank you, guys. Is that happy birthday now? Okay, well, let's go ahead and get started. Let's see. So let me see if I can tell you guys anything I didn't tell you in class. I did go over the final a bit more, probably since the last time I talked to you. So just to kind of recap, I think we were at about 25 questions on genetics and evolution together. So, you know, don't expect it to be in like vast detail. Maybe one or two questions for each of the major topics. So, for example, a question on DNA replication, and maybe 2 or 3 questions on sort of transcription translation-y stuff. A couple of questions on biotech, you know, things like that. It's not going to be some vast amount because, again, it's only going to be 25 for genetics and evolution together. That being said, they'll come back a little bit in some of the integrative questions. There'll be some references to genetics and evolution. But, you know, again, nothing major that if you didn't know most of the big stuff, you wouldn't feel pretty confident about. Okay, and then let's see, I did update some of the physiology sections. So there is still a good chunk of digestive, and hold on, I'm actually trying to pull up, should have done this beforehand, but let me do it right now real quick because I think it'll help you out. I'm just going to pull up the new percentages and then I can... Okay, here's the update. It was 25% genetics and evolution, 30% digestive, respiratory, circulatory, immunity, all of those things that kind of made up that the main part, the first main parts of physiology. That's about 30%. Then there's a section that's excretory, reproductive, and sex ed, and that's about 25%. So I happen to remember it's about 5 excretory, about 10 reproductive, sort of anatomy and physiology, and then another about 10% from sex ed. And then there's 20%, so a good chunk at the end, that's built around integrative questions. So there's a few that sort of tie systems together around homeostasis. And then there's a kind of a whole series of questions that bring in things from across the course, right? So there could be a topic. I think one of them is on, you know, stress or something like that. And it brings in like multiple concepts from across the class. And probably, you know, emphasis still on physiology, but it brings in some genetics and evolution as well. And of course, a bunch of cell bio sprinkled in there. So, you know, nothing too different from what I said in class, but hopefully that gives you a little bit better idea of what to expect. No Punnett squares specifically. So, but some of the terminology from genetics 1 and 2, I mean, you should know the different patterns of inheritance. So things like sex-linked, dominant, recessive, you should feel comfortable with those terms. Certainly genes, alleles, things like that. But again, it's not hugely emphasized. A little bit on gene expression, you know, some of the later stuff is in there. And again, integrative Julie can be integrated across all different units. I mean, overall, the test is probably still about close to 75% physio, maybe not quite so much if you add in the integrative, maybe 2/3 physio. So, you know, definitely feel really strong in physiology, and that will help a lot. But otherwise, you know, again, be prepared for the big concepts and other things. No short answer questions, Jerry. Last thing I want to be doing this week is grading short answer questions, right? So remember, just like the first semester, it's going to be 100 multiple choice Scantron. Captain Bajendra, whoever you are. Well, there is no function of the urogenital papilla in humans because we don't have one. We're not even sure exactly what it does in the pigs. We think it has something to do with mating, and I haven't watched pigs mate enough to know the answer to that. Should we know specific functions of hormones? Some, yes. Certainly the ones that we emphasized a lot in class that we hit multiple times. And there's a couple basic questions about sort of hormone function. And that you might want to refer back to the endocrine chart, where it gave kind of a nice general description of endocrines and endocrines versus the nervous system. Hi, Anshul. Difference between transcription factors and epigenetic tags. You know, we went over this idea that the way that they work is pretty similar in the sense of sort of turning things on and off, especially the process of transcription. But it was a little bit of a difference in timing, right? This idea that transcription factors are sort of what gets used all the time, like day in, day out. Every second that you want to make a protein, you got to attach the transcription factor. Epigenetic tags tend to be more across, you know, the life of the organism. So to get cells specialized in the first place, to make major changes, turning genes on and off that may last for years or the rest of your life, things like that. It's a little more medium term, not evolutionary term, but a lot faster or a lot longer term than, say, what the transcription factors are for, which is literally like, I want to make this protein right now. Okay, but the way that they work is very similar. And again, for purposes of the final, I think there'll be one question on gene expression, so just be comfortable with the basics. Hi, Sathika. What does it mean for DNA to be anti-parallel? Why is it important that DNA is anti-parallel? So anti-parallel just means that the two strands that are complementary, if you look at the actual shapes of the nucleotides, they're actually like this in terms of the way that the nucleotides are bonded together. So, for example, if the sugar is on the top on this one, the sugar's on the bottom on this one, right? And it just has to do with how they're able to bond together to make a double strand. Not really that critically important for what we do in this class, although it's important for how DNA works, right? So it's not something that you need to know for purposes of the final, but just know that, you know, molecules have to be bonded together in certain ways to function. It also is important because it allows your molecules that have to work with DNA to be able to tell which direction to go, because the molecules are literally laid out in opposite directions on either side, so that, for example, if a transcription factor has to land on one side of the DNA in order to cause transcription to happen, it knows what side to land on based on which direction it's going. It's like reading a book from left to right. Hi, Pallavi. Can you explain when the kidney reabsorbs more or less water? That's going to depend a lot on your blood volume, right, which is the amount of water in your blood. So there's a number of things that can trigger it. The obvious one is going to be dehydration. Right? If you're dehydrated, then your kidney is going to want to hold on to more water in your blood, right? Keep your blood volume up. So you're going to want to have more water go back into your blood as opposed to out in your urine. So that would be kind of the most obvious ones. But remember, you should know something we brought up about the idea that alcohol blocks that, so that instead of reabsorbing that water, you'd let that water go through the urine and potentially become more dehydrated. You should also know the connection to blood pressure, that the more water that's held in your blood, the higher the blood pressure is going to be, okay, as opposed to letting more water go in the urine, which is going to lower the blood volume and lower your blood pressure. Do we need to know the parts of the small intestine? I think you're referring to those sort of Latin names like the duodenum and the ileum and the jejunum, I think, if I have that right. No, we don't do that. We mention the duodenum once in a while because it's kind of where all the major stuff happens that we study in terms of all the enzymes coming in and that sort of thing, but it's not something we focused on in this class. Maybe just about villi and why the small intestine is so long. Yeah, I see your point, Pallavi. Yeah, that we studied more about the structures inside of the small intestine and the fact that they needed to be long in order for all that diffusion to take place. Evelyn, is hepatitis viral or bacterial? It is viral, hepatitis virus. Okay, that's why we don't have a great way of curing it, because viruses are so much harder for us to cure. Hi, Hia. Good to see you on here. Let's see. Neo, should we know the phases of meiosis? Nah, you didn't need to know that at the time. You certainly don't need to know it now. You should know the purpose of meiosis and the fact that the cells have to divide twice, right? But we kind of let go of those specific phases. You should have a vague idea of sort of crossing over has to happen in the beginning before the homologous pairs separate, that sort of thing. But we didn't get into the names and what happens in the different parts. We may have mentioned prophase 1 a few times because that's, again, a particularly important section, but not that critical. Bhavna, do we need to know the genetics material from the end of first semester too? Again, I did answer that briefly before, the idea of vocabulary being the most critical. There is a question on meiosis, which I believe is from first semester, because it's just an important, basic concept in terms of reproduction and passing on the genetic material. So you should know, one question about the basics of what happens in meiosis and why it's important. Let's see, homemade chutney, that's a cute name. What are the cancerous STDs? Well, you want to, always try to make connections, right? One of the reasons that some things in general cause cancer is because viruses, which are the ones that can cause cancer, can insert themselves into our DNA, right? So you're going to already narrow it down to STDs that are viral. It happens to be the ones that infect certain parts of the body that are particularly prone to this. So hepatitis is connected to liver cancer, and HPV is connected to virtually all cases of cervical cancer and other kinds of reproductive cancers as well. So penile cancer in males, vulvar, something like that, cancer in females, etc. So if the virus infects any of those cells in the reproductive structures, then they could potentially cause cancer. Satvika, what is an aneurysm? So an aneurysm is the name for the bubbling out part of the artery. So when it's weak on one side and balloons out, then that thing is called an aneurysm. It may or may not have a rupture, right? It just is a name for this sort of bubbling out of the weak wall of the artery. If it actually ruptures, then we would either call it a ruptured aneurysm. If it's in the brain, then we call it a hemorrhagic stroke. Okay? So, but the aneurysm is just the bubbled out part. But we would say that, a hemorrhagic stroke most often is caused by a ruptured aneurysm. Not always. I think arteries can just bleed for other reasons as well, but that's a major cause. Hi, Tiffany. Wondering if we needed to know about the taxonomy of humans and human lineage. Again, the super basics, right? Again, there'll be like 1 or 2 questions related to human evolution. So you should have the general idea of the things we talked about in terms of our connection with other primates, particularly our closest relatives like chimpanzees, right? There is a question about, not exactly what you asked, but there is a question about cladograms as well. So, be prepared for the basics. And I'm just going to I'm looking at another screen here to kind of double check if there's anything I'm forgetting. Okay, there is kind of one basic question about human evolution, which, remember, we connected to some particular traits, and a little bit about sort of geographically, like the fact that we now know that humans evolved in Africa and had these waves of migration out of Africa and that sort of thing. Oh, thank you, Pallavi. Nice description of an aneurysm. Oh, you didn't have to retract that. It was nice. Okay, Henry's asking something about mRNA moving from the nucleus to the ribosome. Good question. I believe that, I don't think it's randomly diffusing. I feel like it would probably need to be more directed and more efficient than that. There might be some kind of little chaperone proteins Some things are sort of latch on to another molecule and get them to move where they need to go. Honestly, I don't know for sure. I mean, I'm sure it's probably known at this point. It's not important enough for me to look up at the moment. But there's probably, I would guess, a molecule that helps it. Also, a lot of the ribosomes are located pretty close to where they can, and the endoplasmic reticulum is kind of wrapped around the nucleus, so they don't have to go very far. So it could be partly diffusion, or it might be being guided by a molecule. Those are sort of the two main possibilities. I'm not sure which in this case. DT, does crossing over occur before or after meiosis 1? Well, when would it have to occur, right? If crossing over is when you have genetic material between homologous chromosomes, like your daddy's number 5 and your mommy's number 5, right? At the end of meiosis 1, those two chromosomes aren't in the same cell anymore, because remember in meiosis 1, the two homologous chromosomes go into separate cells, right? So it would be impossible after meiosis 1 for crossing over to occur because those chromosomes aren't even together anymore, right? So if you forget something like that, just try to work back and use logic it through and just kind of figure out what you do know and see if you can apply the answer to that, okay? So it has to happen during the very first part of meiosis 1. Raga's asking about an increase or decrease in electrolytes contribute to the reabsorption of water. Well, I guess you could think of it a couple of ways, and it's probably more complex than we need to get into here, but you could imagine if you have more electrolytes, that's going to tend to cause water to be retained, right? If you have more salt in the blood, for example, that's going to tend to cause more water to be retained in the blood because of osmosis. That being said, it's more complicated because your kidneys can move the salt and your kidneys can move the water. So in any given situation, it might have to do with whether you have enough electrolytes for other purposes, whether you have ADH, telling your kidneys to keep water. It probably depends on a bunch of other factors. But in general, the important thing you need to know there that relates to homeostasis is this idea that you need to keep your water and your electrolytes in reasonable balance in your body at any given time. Jerry, the parts of the nephron? No, not really. Not for these purposes. That's kind of too detailed. You should get the basic idea that molecules can move between the nephrons in the blood, but it's not going to be about like very specific things, okay, like the loop of Henle or anything fun like that. Different species of humans like Neanderthals. Not too much, Satvika. Nah, it doesn't go into detail about our other relatives. Now, like I said, the only question that's on human evolution is fairly generic about the process in, across time in general. It's not really comparing the different organisms. Maybe a little bit about humans and chimpanzees. I believe there's a question about that. Pallavi, are most of the questions technical or situational? Ooh, that's a good question. A little bit of each. Because it's multiple choice, it's a little easier to get those sort of pathway and organ questions in there. That being said, I try to put some in context because it's just more interesting that way. So especially in the integrative questions, there's a little bit more scenarios, and then that allows me to sort of pull in some different things. But in the main sections, it's a little bit more technical, using your word, a little bit more sort of factoidal. But some things are in context, so there'd be some disorders that we talked about. I believe there's one that brings in anemia and other kinds of things like that. So, definitely if there's disorders that came up multiple times, you might see them pop up on the final for sure. How does CRISPR locate the gene that it is trying to edit? Does it have some sort of RNA or does it locate a specific promoter? Yeah, CRISPR is kind of preloaded with a little section of nucleotides that it then matches to the section that it's looking for, right? And that way it can find it very precisely. So remember, it's a super precise editing system. So it actually has a sequence that's part of CRISPR that you can load in there, that humans have learned how to load in there. Before that, bacteria were doing it to identify, it was part of the bacterial immune system to identify sequences that had previously invaded that bacteria so that they could be fought against, right? But in this case, we've learned how to preload it with specific sequences that we're looking for in the DNA. So it does have a sort of guide sequence. DT, what could potentially happen if there is too much water in your blood? It would cause your blood pressure to go up. You guys have a sense now, I hope, that having high blood pressure is a really bad thing over time. So if you had too much water in your blood all the time and your kidneys weren't doing a good job of bringing it down for a variety of reasons, then yeah, that would cause you to have high blood pressure over time, which wouldn't be so good. Evelyn, are both red blood cells and white blood cells produced and broken in the spleen? Well, think about that. Are blood cells produced in the spleen? Don't think so. All blood cells are produced in a single place. Where are all your blood cells produced? Hope you know that. We'll see if somebody chimes in. But yes, the spleen does a lot of sort of, storing, keeping track of, breaking down, recycling. Yeah, Nao was pointing out about the red blood cells being recycled there. And I think the liver helps out with that as well. You do have a lot of white blood cells dividing and multiplying in the spleen, but it's not where they're originally produced. But a lot of the immune cells can multiply in the spleen. If you're having an immune response, for example, they'll multiply in the spleen. Mr. Chutney is back, or Ms. Chutney, I don't know which, or neither. What are MCRAs used for in terms of evolutionary descent? So that's most it's MRCA. That's why I was having trouble there. My brain was not working on that. It's MRCA, most recent common ancestor. Okay? And it's just a way for us to point out, if you have two organisms, where they were most recently connected, where they had their most recent ancestor. And if you're looking at a cladogram, that's going to be where those two lines come together, right? So at the V is going to be the most recent common ancestor. And sometimes we want to know that in terms of figuring out that they are most closely related to each other compared to other organisms. Sometimes it tells us how long they've been evolving separately, things like that in terms of understanding the evolutionary relationships between things. But it is most recent common ancestor. Go back and forth there. Okay, thanks, Paula B. Okay, somebody else got that too. What is the difference between gender and sex? So, lots of people use them interchangeably, but it's kind of clear if they don't, because they're not the same. So sex would be referring to biological sex, so that could be all the things we talked about in sex determination. It could be your chromosomes, your hormones, your parts, inside and out, all that stuff that we think of in terms of biologically what makes you male or female. Understanding, of course, that it's not completely binary, right? That there's a fair number of people that are not fitting into our standard definitions of male and female. Gender has a lot broader meaning. So gender could be related to gender identity, how people feel, but it's also a term that's used sociopolitically in terms of how we think of males and females in society, masculine and feminine, right? And in terms of all the issues that arise. You could talk about gender equality or something like that. So that has a lot more different kinds of meanings than sex. Okay, sex is going to be much more around biological maleness, femaleness. Gender can be personal, social, political. Some people will talk about gender being a social construct, right? Because we sort of decide on these male and female roles and that sort of thing. So it'll be have a lot more different kinds of meanings. Sathika, what types of molecules are actively secreted during secretion and why? I think you're referring to the kidneys. So in general, it's going to be things that are larger, so it could be whole proteins, for example, that wouldn't go through during filtration. It could be things that have to be actively transported. So usually, like if ions are going against the concentration gradient like that, that doesn't have to be big. That could be something like hydrogen ions if you're trying to adjust the pH of the blood, but you may be having to actively transport them if they're not going with the concentration gradient. So then you would do it during secretion. Okay. Do we need to know about the new kingdoms that originally were part of the Protista kingdom? Wow, Anshul, no way. Whew, that'd be way too much, because there's like a whole bunch of them now. I don't know them all. Yeah, back in the day, you could know all the kingdoms. We're long past that. There's like 50 or 60 or more kingdoms now. I can't even keep track. So no, you don't need to know that. I'm glad you know that Protista is no longer a kingdom. It's been a while now, but not everybody's even aware of that. So no, I wouldn't expect you to know that sort of thing. Nusha, is there a question on cladograms? You bet, there's an actual cladogram. Ooh, picture, right? So there's going to be a picture, a sample cladogram, and you're going to answer some questions similar to the ones we worked on in using cladograms. So, being able to interpret how things are related, being able to say which organisms share a particular trait that's labeled on the cladogram, things like that. Again, all the same stuff we practiced on using cladogram. Like there might be some general question about what they're for, right, and what they indicate. Hey, Nick. Blood cells are produced in bone marrow from stem cells. You got it. Those hemopoietic stem cells. Thanks for chiming in on that. Okay, and that includes all blood cells. Red blood cells, white blood cells, the kinds of blood cells that give rise to platelets, etc., etc. Henry, do the veins have smooth muscle or skeletal muscle? It's a good question. But again, that's one you could use logic for, and I'm just going to keep encouraging you to do that because, inevitably there'll be something on the final that you didn't necessarily prepare for. So think about that. Skeletal muscle implies that they're attached to your skeleton, right? So those are going to be all the muscles that make your bones move, your arms, your legs, your ribs in the case of the diaphragm, right? If they're making movement happen because they're moving a bone, that's skeletal muscle, okay? All the involuntary muscles that are around soft, squishy things, like around your stomach or around blood vessels, those are all going to be smooth muscle, okay? And they're generally, not always, but often involuntary, and they are not attached to bones. Okay, is there white bone marrow and red bone marrow? Yes, there's also something called yellow bone marrow, which has more to do with fat. We didn't really study those a whole lot. Mostly we focused on the red bone marrow, which is the part that makes the blood. How can we determine which organism is closely related to another organism? Well, it's all relative, right? So, Frank, it's going to depend what you're looking at, right? Remember that the main concept is to look for the most recent common ancestor, okay? I don't have a paper right now, so I'm going to try to make this with my fingers, okay? So, let's see here. These two, on my one hand, they're going to be more closely related than either one is to this one, right? Because these, over on this hand, the ones that are wiggling, share a recent common ancestor more recently than they do with this one, whose recent common ancestor is down here. Okay? So if you can look for the one with the most recent common ancestor, that's going to be the ones that are most closely related. So, look for that recent common ancestor. The ones that share a common ancestor most recently are more closely related. So I just had my most able assistant bring me a paper here. Okay, so let's see. We'll call this, let's see. I think we'll call this A, B, and C. Let me draw something, names on here. A, B, C. There we go. Hope you can see this. Not sure you can, actually. Put that in front of there. Didn't show up very bright. Let me go over that with pen. Don't have my whiteboard today. Okay, we'll see if that's a little bit better. Sorry, I'm waiting for the picture to come up so I can see it. Okay. I'm not sure if you guys can see this or not. But anyway, these two here share a more recent common ancestor right there. Ooh, yeah, see, I got to get this in the right angle with the light. Sorry, a little technical difficulties here. We're doing this the old-fashioned way. Okay, so here you've got B and C, and you can see that they share an ancestor here. More recently than either one shares it with A. Okay, so the most recent common ancestor of B and C is located right here. The most recent common ancestor of all three is down here. But that means if this is some point in time in the past that these two diverged, speciation, right, and that these have been evolving on their own pathways, and then after a while, these two diverged, right, and they started to evolve on their own pathways. But the implication is this was evolving on its own pathway for a longer time than these two, so it had more time to accumulate more changes, right? So B and C would be the most closely related. A would be slightly more distantly related. So this would be like you and your sibling and both of your siblings and your first cousin, right? Your parents, your grandparents, okay? So sharing that most recent common ancestor means you've had less time that you've been diverging from each other and becoming more different. If you have to go further back in time, you've had more time to become different, so you're less closely related, okay? And you might want to go back and watch that nice video, if you remember that sort of choose your own adventure video. That was, I think, super helpful about this, and this idea that all that matters is where these lines come together. I could have put A out here, and it wouldn't matter. It's a matter of where these lines come together. Okay, so remember that. And if I put a trait here, it implies that both B and C are likely to have the trait, but not A. So if I put a trait right here where my finger is, that means it evolved here. And likely B and C both have it. Not always, because sometimes maybe B lost the trait, and maybe A gained it. Maybe it evolved independently out here. So it's not a guarantee, but it gives you some idea of how things might have evolved. Okay? So hopefully that was helpful. Okay, Julie's asking, do we need to know all of the water pathways? Are you thinking about from ecology? Nah. No, I didn't go back that much into ecology. There's a couple questions relating particularly back to climate change and certainly back to energy flow. So if you think about how energy flow that we learned at the beginning of the year kind of ties into everything all year, right? In fact, that stuff I do the first week of school, I do that on purpose, right? So if you remember, you probably don't remember, but I remember, the first week of school, we did homeostasis and metabolism as characteristics of life. And those have come up over and over again this year. And then we spent the next week or so after that doing energy flow, right? And that also, again, has come up multiple times throughout the year because those are such basic concepts. I wanted to keep reinforcing them all year long. But in terms of the specifics of water, water treatment plants and such, nah, that's not going to be on this final. Aditi, how does an organism originally have RNA polymerase to produce proteins through transcription and translation? Are you talking about like an embryo or like life in general? I'm going to answer that for an embryo, okay? So RNA polymerase is a protein, right? So if you think about when an embryo is first formed, that it has all the information to make all the proteins it will ever need, right? So it's got genes that code for RNA polymerase. So as those cells are first dividing in an embryo, the genes within even that first fertilized egg cell are starting to make specific proteins. Then some of those specific proteins are causing other proteins to be made, right? So that they're able to get started. Because again, the sperm and the egg were functioning cells with their own DNA, with their own RNA polymerase. It's not like you had to start from complete scratch, right? You started with two functioning cells, a sperm cell and an egg cell. So they already were functioning cells. They already had RNA polymerase. Why? Because they had their own DNA and they had their own ribosomes, right? So they already were able to make everything that they needed. And basically you just continue doing that. You continue making more and more things. So, you don't have to think of it as starting with nothing. And you're right, it would be hard to sort of imagine how to start from nothing. And people who study about the evolution of the origin of life have to sort of think about how did the first cells occur, right? And you basically have to have a much more basic system in which they think actually RNA was perhaps the first molecule that got everything going before there were even proteins. But that's another subject for another day. Do we need to know about blood typing and all that stuff? I'm assuming all that stuff you're thinking of the stuff from genetics, the first part of genetics, right? When we did sex-linked and all that. Again, there's a few basic references. I think there's a couple of questions that use the term sex-linked trait. In terms of blood type, we partly use that not just for blood type, but also to understand a little bit about antigens and antibodies, and that will come up on the final in a couple of places. So, again, some of the basic concepts, but it's not going to be in great detail about, type A and type B and that sort of thing. Hi, Elena. Do we need to know the 4 types of chromosomal mutations? I don't think there's a specific question about like inversion versus translocation. It's not about that. There is a question about mutations in general, right? So you should understand a little bit about the concept of mutations, like which mutations do what and why they might be more important for cells, why they might be more likely to cause changes, or how mutations cause changes in terms of proteins and in terms of how cells function. Also, some of the common and more important mutations that we studied in terms of disorders. Yeah, it's more general like that. It's not so much about, we didn't really get into, even during class, much about the specific chromosomal mutations. You should have a sense that they can occur, right? But we didn't really even talk much about why those were different from each other. Briefly, Satvika, inversion is when a chunk, when a chunk of a chromosome turns like that, which sounds crazy, but it can do that. And then a translocation is when a piece of chromosome breaks off and sticks on another chromosome, right? So like a chunk of number 12 could break off and stick on the end of number 5, which again seems really wacky, but it's not actually all that uncommon. And it's not necessarily that much of a problem. Ooh, looks like Henry piped in with some descriptions there too. Thanks, Henry. Do we need to know the carbon cycle? You need to know it conceptually, yes. You really can't understand life on Earth without understanding the carbon cycle. So again, we won't get into a lot of detail about some of the stuff that was more first semestery, so say, you know, permafrost or something, right? But in terms of understanding how carbon goes in and out of living things, oh yeah, there's definitely a few questions about that. So you should be able to understand, for example, how carbon got into you, how it gets out of you, right? Where does it go? How does it go those different places? Again, that was in ecology, it was in cell biology, it was in physiology. You can't escape it. You need to know how biomolecules work in living things. I mean, that's the basics of biology. So a little bit, yes. Safka is asking about specific STDs. Chlamydia, gonorrhea, HPV, HIV, herpes. Yeah, I don't even think all those are specifically mentioned necessarily, but those are the common ones. You know, the other one people get tested for is syphilis, and of course hepatitis. But yeah, those are kind of the basics. And Frank, I just talked about the carbon cycle. Safka, this came up earlier. Hepatitis is a virus that infects your liver cells, different types. And not even all sexually transmitted. Hep A is in food and water. Victor, do we need to know non-disjunction? Not in a great amount of detail. Remember I said there was one question on meiosis? So you should know the basic idea of meiosis. I don't even remember if that question mentions non-disjunction. I don't believe it does. But, you know, yeah, just in one sentence. Remember, that just means that the chromosomes don't sort correctly. So you could end up with too many or too few chromosomes in a cell after meiosis. And that's plenty to know. Neil's asking, is it a good idea to know specific diseases? You know, I'd say if they're diseases that came up a whole lot, they're likely to end up in a question, right? Because it just puts things in context. So, you know, think in terms of each system. We focused on some really specific diseases. So if we were talking about the digestive system, we did a little bit on, I don't know, acid reflux or something like that. If we did respiratory, we talked a little bit about some of the biggies, like bronchitis and asthma. You know, if we were doing the blood, anemia, so, and, you know, certainly heart disease, diabetes, the ones that came up over and over and over, right? Definitely know something about those, because questions will likely be in the context of some of those disorders. No worries, Frank. Hi, Nick. Into how much detail should we know about the content of blood? You know, again, there'll be like one or two questions that talk about blood. I would say the more things came up, the more you should know. So, you know, we hit fairly hard on things like red blood cells carrying oxygen, for example. We didn't do a lot on blood clotting. There might be like one mention of it. You know, we didn't get into some of the more obscure things. We did talk a little bit about there being things like cholesterol and fat in your blood, right, related to cardiovascular disease. So again, a good judge is just, did it come up more than once? Did it come up in context of something else, right? But other than that, there might be a question about the kinds of molecules you'd expect to see in blood, because I think that's a good general measure of your knowledge of sort of how things are transported around the body. That's about it. Roger's asking about sexual orientation. So that implies who somebody is attracted to, right? It's gotten a little more complicated as we rethink gender, right? Because a person might not even be attracted to a particular gender. They might be attracted to just people regardless of any gender identification, right? So, but it implies romantic or sexual attraction, right? So who you want to be romantically involved with or sexually involved with, and for some people those are two different things, for some people they're the same thing, but as opposed to, say, gender identity or gender expression. Sathvika, are all bacterial STDs curable? Generally speaking, sometimes if they get really far along, the antibiotics aren't that functional anymore. Syphilis, when it's advanced, it gets to a point where the antibiotics aren't going to help that much. Also, there's a little bit more concern now with antibiotic resistance, as you know. So antibiotic-resistant gonorrhea may not be curable with our standard antibiotics. Bobna's asking about the disorders caused by non-disjunction. I mean, the only ones you mean, there's all the sex ones that we looked at. Oh, I think I used to have a question that mentioned one of those, but I don't think I do anymore. We really only looked at the things like, you know, wow, the word escapes me, like XXY males, Klinefelter, and then things like Downs. I don't think they're specifically in the final. No. Safika, how much do we need to know about STDs? There are 2 or 3 questions that incorporate STDs, so you're going to want to know the basics that we went over in class multiple times, so about how they're transmitted and what kinds of things prevent them, what kinds of things don't prevent them, things like whether we can treat or cure them, right? The connections to cancer, so all the same stuff we went over in class. You know, who has STDs, who doesn't, which is pretty much everybody, by the way, that has them at some point or another. So kind of the basics that we hit hard in class. Again, 2 or 3 questions. Hi, Lillian. Do we need to know anything from cell bio? Oh, yeah, right? So if you think about how we pretty much couldn't get through physiology at all, like pretty much any system, without talking about a fair amount about biomolecules, right? So anything that came up second semester is fair game. Plus there's some integrative questions that really incorporate basic knowledge of how biomolecules work in living things. Like, where do they go? How do they get from the air into your body and out again? And, you know, how do they get changed from one molecule to another? There's some questions that ask about enzyme function, right? So anything that sort of puts together the way our bodies work or puts together biology in general, that's from cell bio? Yeah. Because cell bio wasn't just about cells. It was really about all the basic biological functions that make living things work, right? How molecules are manipulated, the kinds of molecules, what happens to them, how they're converted from one thing to another. So yes, significant amount of cell bio. Okay, there's going to be references to diffusion, et cetera, et cetera. Neo, anything about stabilizing directional and disruptive selection? I don't believe so. I want to say not. But certainly a bunch of questions about selection in general, right? So you should be able to apply it to the cases I gave you in class, like antibiotic resistance, for example. There'll be a couple scenarios that you haven't seen before. I want to check something here to remind me. What else there is about evolution? You're going to want to you know, be able to apply the idea of selection to changes in the environment like we did. There is a little bit, there's one question that kind of covers some of the terminology in evolution in terms of comparing, for example, coevolution, convergent evolution, genetic drift, vestigial structure. So some of that, there'll be some examples of some of those things, right? And you also want to be able to apply, and this was something I meant to say when somebody asked about mutations, you're going to want to be able to connect mutations with, of course, genetic variation, right? Because in terms of evolution, that's super important. So a few connections between genetics and evolution as we did in the unit. So remember, a whole big section of evolution was really connecting genetics and evolution. So definitely some of that. Oops, hold on, I lost track of the questions here. Okay, Shannon's asking about examples of metabolism. Let's see if anybody piped in on that one. Okay, so I'm going to answer your question generally, Shannon. Again, I want you to really work through the knowledge that you have, right? You need to know what metabolism is, right? So metabolism is just chemical reactions, that's all. So you should be able to come up with lots of examples of chemical reactions that happen in your body, right? If you think in terms of the things that you have to build, I shouldn't say things, the molecules that you have to build in your body, when you're making stuff, you make all kinds of stuff. Think about the different classes of biomolecules. You're making all of those in different processes. So think about the different biomolecules and why would you build those? When would you build those? Where would you build those in your body, right? In terms of breaking things down, what would you have to break down? Why? When? Where? Right? So I'm going to push you a little bit on that one to think about. The reason I ask a question like that on a review is not because I'm going to quiz you on the types of metabolism. It's to get you to think about the stuff that you know and how it relates to metabolism so that I can throw some examples at you in multiple choice questions. And you're going to already have thought about what all those different things are and why they're part of your metabolism. Okay? So see what you can do with that. Bhavna, other than type 1 diabetes, are there other autoimmune diseases that we need to know? Oh, I think I used to have a question about multiple sclerosis, but I don't think I do anymore because we didn't talk about that much. Most of the other ones we didn't talk about in much detail, so I don't believe, unless I'm using them in a scenario, in which case I would describe them to you. So there's a few scenario questions that might involve things that you haven't heard about, similar to the way I do that in the practice questions, but I would provide enough information within the question so that you'll be fine. It's just to test you on basic stuff, just putting it in some kind of an interesting context. I don't believe there is another autoimmune disorder specifically, not that I can remember anyway. And I certainly wouldn't ask you detailed questions about it. Technically, AIDS is not autoimmune. It is a disease that affects the ability of your immune system to function because your immune cells have been infected. But it's not autoimmune because it's not that your own body has attacked your immune system. A virus has, right? So it's not autoimmune, but it is an immune disease. Okay, so a little bit different. There are immune disease, and remember the term disease is used really broadly. Sometimes it's used for pathogens, sometimes it's used for when parts of your body aren't working right. So an autoimmune disorder is when your immune system is attacking some part of you, right? So that's a malfunction of your immune system. But you can also have outside pathogens attack your immune system, or something else that causes your immune system not to work so well, in which case we would call you immune compromised. Okay. Valves of the heart. No, I don't specifically test you on valves of the heart, at least not the names of them. There could be a reference to how valves function and why you need them, but I said early on that I wouldn't require you to know the names of the valves. You do need to know some basic gender and sexual orientation stuff, and particularly you need to know what those things mean, right? So you should know what we mean when we say gender identity and gender expression and sexual orientation, right? It's not going to be in the kind of detail that was on the matching. That was kind of more for familiarity. So you've seen some of those terms in case you hear of people who identify using those terms, okay? But it's not going to be in that kind of detail on the final. And again, keep reminding yourself that's how it is for most everything, right? There's one question that's on gender and sexual orientation. So it's not, it's not going to be that detailed. Do we need to know the types of energy resources and their pros, cons? Again, that's the kind of detail from first semester that we're not really going to go into. There might be some reference. There's a bunch of questions that tie together stuff around carbon and energy and molecules because that is kind of a theme the whole year. I may have thrown in something in there about there sort of being energy in molecules or energy in certain kinds of energy resources, but not in great detail, not what you're talking about in terms of like the pros and cons of, nuclear versus fossil fuels versus hydrogen fuel or something like that. It's not that kind of detail, no. Okay, how do cells regulate gene expression? That's a really broad question, so hard to answer in the short term. I would say refer back to the gene expression PowerPoint. There is a question on the final that refers to that, right? And we talk a little bit about different mechanisms for turning genes on and off, for controlling transcription or controlling other parts of making proteins. But again, it's one question, so it'll be somewhat more general about the idea that cells can control the expression of the genes or making proteins in different kinds of ways. The treatments of renal failure now. I would say you should know what dialysis is, right? We spent some time on that. I mean, in terms of treatments of renal failure, the only ones we really talked about are dialysis and transplant. So I think that's reasonable to know those two things. But again, not in great detail. There's probably one question that refers to renal failure. Raghu's asking, is the purpose of the villi to make sure that the blood of the mother does not come in contact with the blood of the fetus, so blood type issues do not occur? Yes, generally speaking. It wouldn't be just blood type issues, though. You'd have a bunch of immune incompatibility that could occur. It also is to prevent not just the interaction of the two immune systems, but also it helps a little bit with decreasing pathogens because bacteria are generally not able to go across the placenta. So you also get some protection against pathogens that might be circulating in the mother. It actually keeps some of them from getting into the fetus. Should we know any of the gender and sexuality type of stuff? Yes, as I've already described, you need to know the basic ideas of gender and sexual orientation. What type of protection would be used to protect against STIs? Well, in class we talked a lot about two major things, which is direct protection, which would be like a barrier, like a condom. So you should have some idea of what that can protect against, right, and what it can't, knowing that condoms don't protect against everything or provide sort of partial protection against certain things, right? They're best for protecting against semen getting from one body to another, right? Because the semen can't go through the condom. They're not so good for protecting against anything that can be on skin, right? So many of the viruses, for example. But you can also think in terms of that question a little bit larger, right? That part of what protects against STIs is people communicating with their partners, people getting tested on a regular basis and communicating their test results to a partner, right? Being responsible about your relationships and your sexuality. So there's that kind of larger level of protecting against STIs as well. Are allergies autoimmune diseases or the response to the disease? Allergies are a sort of overreaction of your immune system. So remember that business about the sort of homeostasis in your immune system, not too much tolerance, not too much reactivity, right? So it's this idea that, when your immune system was developing, for reasons we don't entirely understand, my favorite phrase in biology, that you might become hypersensitive to certain kinds of allergens, certain types of antigens in the environment, and therefore have a big immune response to them when that's not necessary, when they're not really going to hurt you, right? And so that's what we're talking about. So it's not an autoimmune disease because it's not your immune system attacking yourself. It's still attacking other things that are not yourself, but it's attacking them inappropriately. So that is another problem with the immune system, but not an autoimmune disease, right? So when we looked at immune disorders, autoimmune diseases is a type of immune disorder, allergies is a type of immune disorder, HIV is a disorder that occurs in the immune system, but those are all separate things. Bhavna, do we need to know about patterns of heredity? Codominance and complete dominance, etc. Not much. There may be one or two minor references to it, maybe. More in context of other things. I'm going to just flip right now and quickly check and see if I'm forgetting some reference. Yeah, not really. I may be forgetting some minor reference, but no, again, mostly the stuff from the first part of genetics is very general. Again, there is one question about meiosis and a little bit of terminology, but nothing specific like that. Are most of the questions going to be similar to the ones on the review sheet? Not really, Anusha. I mean, the questions on the review sheet were to get you to review concepts that will be in multiple choice questions. So a lot of those questions on the review sheet aren't directly the kinds of things that one could easily put in a multiple choice question, right? But sort of make connections. So I gave you an example before that there was this general question that was like, give some examples of metabolism. I'm not going to say give examples of metabolism because that I can't put into a multiple choice question very easily, right? But I might say which of the following are accurate examples of metabolism, right? So it could be testing that you know what metabolism is, but also are the specific examples accurate? So use those as a guide. I wrote those questions specifically for concepts on the test that people struggle with, right? But they may not translate into the exact wording of a question, right? But they'll definitely put you in the right direction. How many questions are there about nutrition? I would say 2 or 3. There's one that's very specifically about nutrition, but that nutrition could also be referred to if you think about, I would say you could certainly expect 1 or 2 questions about cardiovascular health, and that could incorporate some nutrition, right? So I'd say 2, 3, 4 might have references to nutrition. Out of those, there's one that's like very specifically about nutrition, but maybe a few other references. Ting is asking about the purpose of splicing. So splicing allows you to use a gene in different ways, right? So you don't have to go with the exact order of the nucleotides that are in the original DNA. You could cut pieces out either because it provides a protein that functions better or differently, right? And it allows you to potentially use that same gene to produce different proteins, which is actually quite common. That's alternative splicing. But really, it's just, there's a lot of things that are part of how genetics works that really just generate more variation. So splicing allows you to get more variation of proteins out of a common sequence of DNA. And you may not use all of it at one time, but it allows that variation to be there, right? Which just allows us to be really efficient with sort of a small amount of DNA. Is the review sheet everything on the final? Wow, that's asking a lot, Victor. Of course, it's not everything. No, there's no way to do that, and it's not helpful, right? Again, the purpose of the review sheet is to point you in the right direction for major concepts, right? And to maybe get you to review a few things that you may overlook without that, right? That I know are on the final. But, you know, there's going to be some details that aren't on that are details of concepts for sure. And there's a few little things in there that are just in one question that I didn't put in a review sheet because it would make the review sheet too long, right? So, but certainly it will point you to most of the major concepts that are on the final. Making a hello, I don't know you, but welcome. Why do cells go through gene expression? Well, if you think in terms of gene expression really just being protein synthesis, why do cells do protein synthesis? Because they need to make proteins. Why do you need to make proteins? Because they are most of our bodies, because they are enzymes that do things, because they are you know, molecules that transmit things and transport things and send messages. They pretty much do everything, right? Other than being a major source of energy, they're even a minor source of energy, right? So proteins are critical for life. So if you think in terms of gene expression being basically making proteins, hopefully that helps you with that concept. In terms of why you might be asking, why do we need to regulate gene expression, which is another question. But you can connect it to the first question. Proteins are so critical, of course we need to control which proteins we're making at any given time, because proteins make things happen, right? So, and proteins rebuild parts of us. We don't need to rebuild parts of us all the time. Sometimes we need to make more of this and less of that. Sometimes we need to make this reaction happen and not that one. Sometimes we need to send this hormone and not that one. So we constantly need to adjust which proteins we're making in any given cell, right? What does that cell need? What does it need to make? What does it need to do? How is it responding to the environment? So that we need ways of turning on and off genes. Maybe it's because the cell is specializing or only has to do a certain job with certain proteins. So we need ways of controlling what proteins are made at what times and what cells and how much of them. So that's why we need to regulate gene expression. Hope that answers your questions. Okay, Tiffany's asking, is it correct to say that body cells are diploid while reproductive cells like the egg cell are haploid? Okay, so the issue with that question is that reproductive cells is such a vague term that I don't know which reproductive cells you're referring to. That's why I'm going to steer away from terms like reproductive cells or sex cells. I'm like, what does that mean exactly, right? I can't even tell you what that means because that's a meaningless term. I think you might mean germ cells, but then you're referring to gametes, which are two entirely different things, right? So if you think about most of your body, including your reproductive organs, is made of diploid cells, right? The only haploid cells you have are gametes, okay? But the cells that make the gametes, the cells that go through meiosis are germ cells. They are diploid. By definition, a diploid cell, 2N, that goes through meiosis will produce 1N cells, your gametes. Okay, but when people use vague terms like sex cells or reproductive cells, I don't know if they're only referring to the gametes or to the cells that divide to form the gametes. That's why I don't like those terms. It's more precise to use terms like germ cells, somatic cells, and gametes so that we know what we're talking about. Okay, but certainly most every cell in our body is diploid, including our germ cells. The only cells that are haploid are going to be our gametes, the actual eggs and sperm that we produce. Let's see, does meiosis 1 produce haploid cells or diploid cells? Those are haploid cells, right? Because already in meiosis 1, the two homologous chromosomes, one from mom and one from dad, they already separated. So instead of having 2 copies of chromosome number 5, I already only have 1 copy of chromosome number 5 in each cell. It's just random which parent it came from. But there's only 1 of that type now in those cells. So already at the end of meiosis 1, I have 23 chromosomes, 1 of each type, right? So those are haploid cells. But each of those chromosomes still has 2 sister chromatids, because those don't split until meiosis 2. Okay, we're in. I'll go a little while longer tonight. We'll see how I'm feeling. Whew. Kind of tired. Long weekend, lots of fun. Okay, and you guys shouldn't stay up too late either. You got 2 finals tomorrow. Lots of you in my class. So make sure you get a good night's sleep tonight. We'll cut off pretty soon. What happens to the introns after splicing? They're gotten rid of. I assume they're recycled, broken down. You know, they're just strings of nucleotides, right? So you can just break them down. Your cell could reuse the nucleotides. It could break them down completely and excrete them and get rid of them. Not sure. Those are pretty much always the options, though, right? Recycle it or break it down completely and get rid of it. Introns exit. Thank you. Dang, that is so true. That's because they're interrupting or intervening sequences. Where do they do? I'm not sure of your question, Frank. Safika's asking, are you born with epigenetics or are they permanent? I think you mean epigenetic tags. Okay, epigenetics is like the name of the whole concept. You are born with some. We're still trying to figure this out. But remember that a lot of epigenetic tags have to do with cell specialization and gene expression in your cells, so that sperm and eggs are going to have some specific epigenetic tags that have to do with them being sperm and eggs, right? And in order for those to become embryonic stem cells, which can then become anything, a lot of those tags have to be cleared off. So there is a generational thing that happens that at some point between having sperm and eggs and having an embryo, a fertilized egg, in that very early stage, a lot of those epigenetic tags are cleared off so that you can kind of start with a clean slate, but not all of them, because we do know that some epigenetic tags are passed from generation to generation. That's what's so interesting about this. We don't know why some stay and some don't, right? There seem to be some that are attached, for lack of a better term, very strongly, right? Particularly when they're associated with sort of trauma in previous generations, and those appear to potentially stay and affect gene expression in the following generations, right? But most of the sort of ones that, for example, would make a cell become bone cell or a nerve cell, those are removed because you have to have the cells be unspecialized again in order to make a new embryo, okay? So many of them are removed, but some are not. Okay, let's see. Bhavna, in terms of your question, the main concepts from each section, honestly, that's what those final exam review questions are for. I literally went through the final and put the major concepts into those final exam review questions. So I don't want to list them in this live stream. I don't think that's helpful, but please go back to your final exam review and those questions I listed. Literally, I made that list looking at the final. And in fact, I updated it when I redid the final for this year. And if you guys didn't get the message, I sent out an email, but I did update the final exam review questions, and the new version is posted on School Loop in the top part of the locker. It's not like vastly different. Mostly I updated some stuff related to sex ed and took off some things related to the nervous system because those won't be on the final. Would it be a good idea to know how to pair AGCT? Yes, it would, Nao. You will be doing that in one question. Raghav, just to make sure, homeostasis is an example of metabolism, right? No, it's not, Raghav. Okay, so that came up early in the year. Is homeostasis the same as metabolism? It's not the same, and it's not even really an example. Metabolism is specifically chemical reactions. Now, in terms of maintaining homeostasis, do you sometimes do chemical reactions? Of course you do, right? For example, if I'm going to maintain blood glucose, I'm going to sometimes build glucose into glycogen and sometimes break down glycogen and release it as glucose to maintain homeostasis of blood sugar, right? But maintaining that homeostasis, keeping something level or within a certain range, is not metabolism, because metabolism is doing chemical reactions, right? So there's lots of things I can maintain, for example, blood pressure, without doing much in the way of chemical reactions at all. And there's lots of chemical reactions occur, like breaking down my food and digestion, that don't have anything specifically to do with homeostasis. So those are really separate concepts. What holds them together is they're both characteristics of living things, right? They're both really basic concepts in biology, which is why they both came up early in the year. But they're not the same, and one is not an example of the other. Safika, do we need to know the different phylums and kingdoms and classes and so on? You know, if you think again back to this class, we didn't do that a lot, right? I wanted you to know the order in terms of understanding that it's a nested system, that one is inside of the others, right? You should be able to relate that to cladograms. You should know some of the very basics in terms of like life, like bacteria versus, eukaryotic cells and animals and plants and that sort of thing. But we really didn't go into a lot of detail, right? We did a little bit with human beings particularly, but that's really, was kind of more for interest because we're humans. But, you're not expected to know much of that. You should be able to use those words in context if they come up. Bhavna's asking, can multiple alleles affect a trait, or is it always just two? Nope, multiple alleles, and in fact, most of the time, multiple alleles affect a trait. Most traits are polygenic, remember, so there would not only be multiple genes, but each of those genes could have multiple alleles, right? So we looked at blood type as an example. That was just 3 alleles, A, B, and O, that can affect the phenotype. But actually, there can be hundreds or thousands of alleles. If you think about the fact that a gene is often hundreds or thousands of nucleotides long, and over the course of time, one person or one organism has a mutation here, and another organism has a mutation here, and here, and here, and here over time, there can really have hundreds of alleles. It just means a different sequence of DNA for that particular gene. So it's very common in any given, let's just use humans as an example, in any given human gene, if I compare my gene to yours, we probably have some different sequences, right? That's why when we compare human genomes, that we're all a little bit different, because we've got this genetic legacy of these different mutations over time. They don't necessarily make the gene work differently. Sometimes they do, right? That makes some of our variation. But certainly there are many, many alleles of every gene. Not every gene, but most every gene. And for most every trait, there are multiple genes that affect that trait, right? So most traits are polygenic. What is the difference between gametes and germ cells? I went into that at some length before, but just to answer that question briefly, germ cells are the diploid cells that divide to form the gametes, which are the sperm and the eggs. So you have a bunch of germ cells in your ovaries or testes, generally speaking, one or the other, whose job it is to undergo meiosis to form gametes, right? So those are two different types of cells. Thanks, Pallavi. Got that pairing down. Mira, hi Mira. I haven't seen a question from you so far. Just checking that there won't be specific questions on eco-cell bio, but there'll be questions that connect concepts. Yes. And anywhere where they came up in context previously, certainly I would expect to see that. So for example, we spent some time thinking about the impact of climate change in terms of evolution. Ooh, lo and behold, there will likely be a final exam question on that. In fact, there will be. So in terms of those sorts of things. Otherwise, they'll just be passing references that connect concepts, yes. But, again, expect to see a fairly heavy presence of cell bio. And actually, there are a few specific questions on cell bio, now that I think about it. So, for example, there'll be a couple specific questions on enzyme function, because you really can't understand physiology without understanding about enzyme function. Bhavna, what is the purpose of epigenetic tags, and do we need to know which chemicals they're made of? Let's see, the second part, no, you don't need to know which chemicals they're made of. There's a couple common ones, but you don't need to know them. And the purpose is related, again, I went over it at some length before, this idea of regulating gene expression. So epigenetic tags are a way of regulating gene expression, usually over the longer term, in terms of specializing cells for the rest of your life, or making major changes to gene expression in response to significant experiences in your life, right? So trauma might cause genes related to stress response to be different over the course of your life, things like that. Okay, Raghu, so asking about gender identity and gender expression. So gender identity is, generally speaking, the way you feel about what your gender is. So like, do you feel more masculine or feminine? Do you feel more male or female? What do you identify as, right? Maybe you don't feel like either one, and maybe you identify as non-binary because you don't think that the idea of having male and female applies to you so well, right? Or that it shouldn't exist at all, right? So those are gender identity terms. Gender expression is literally how you express yourself. Behavior, clothing, speech, activities that you like to do, things like that. And so that ties in a little bit with gender norms in terms of what people expect in society, but it could also just be an internal feeling you have that you want to express masculinity or femininity in a certain way, and those terms can apply to either sex or people who are non-binary, right? So people who identify as female can still express a sort of masculine part of themselves, and vice versa, right? So those terms are different, and you can see a nice written description of those terms on your gender sexuality matching. Okay, so you might want to refer back to that. Do all genes code for proteins, Bhavna? No, they do not. So genes in general, first of all code most directly for RNA, right? Because remember transcription and translation, that genes are transcribed into RNA. That RNA, if it is mRNA, can then be translated into proteins, okay? So many of our genes, but not all by any stretch, right? Many of our genes eventually are transcribed into RNA and then translated into proteins. But a significant number of our genes are only transcribed into RNA, and then that RNA has a separate function, right? Maybe it's tRNA and it helps with translation, or maybe it is siRNA and helps with regulation of gene expression, right? So we make lots of kinds of RNA from genes, because that's how you make RNA, by transcribing a gene, right? So we make many kinds of RNA that have many kinds of functions. Most RNA in general does relate to regulating gene expression. That's kind of its most common thing. And of course, a big chunk of it is mRNA, which could be translated directly into proteins. Not directly, but eventually. Let's see. Do we need to know disorders or diseases? So, Liliana, I addressed that one earlier in the live stream. Your question's really vague, so I don't know if you're talking about genetic or physiological or in general. And what I said is that similar to this certainly to the entire physio unit, but even throughout this year, we used a lot of disorders and diseases as examples to understand how things work. So if we talked a lot about bronchitis or anemia or diabetes during the school year, you can imagine that those are going to come up, right? Cardiovascular disease. So all the major ones that came up over the course of physiology, yeah, you're going to probably see those on the final as well. Not super obscure ones that we didn't talk about a lot, but certainly the basic ones. What was the protein that unzips the DNA? That was helicase nail. There is a question about DNA replication, but it's like one question. It won't be like vastly detailed. Oh, thank you, Sonic. Victor, does a gene code for a single protein? Or do different alleles of a single gene all code for different proteins? Okay, let me slow down and read that one again. Does a gene code for a single protein? No, a gene can code for multiple proteins. That I talked about that briefly with splicing. Or do different alleles of a single gene all code for different proteins? Okay, well, that's going to depend. First of all, in any one person, unless you have trisomy, let's just let that one go. Let's say you're an average person who has 2 copies of each of your chromosomes. Those could be different alleles, in which case they could code for different proteins, right? You'd be heterozygous. In terms of different alleles in different organisms, like me and you have different alleles, yes, they could code for slightly different proteins. But they don't have to, right? Remember that not all changes in DNA code for different proteins. You could have silent mutations. They don't necessarily code for different proteins. They could code for slightly different proteins, but it doesn't make that much different in our phenotype. Or they could code for different proteins that make a difference in our phenotypes, right? So all those things are possible. What is the difference between proteins and enzymes? Are proteins enzymes or vice versa? Enzymes, by definition, are proteins. Okay, so enzymes are biological catalysts. Just means things in biology that make stuff happen that are made of protein. They're not the only catalyst. RNA can make things happen, but it's not a protein, so we don't call it an enzyme, right? Ribosomes is like this super molecule that contains RNA and proteins and acts like an enzyme in terms of making things happen, but because it's not solely made of proteins, we don't call it an enzyme, okay? So all enzymes are proteins, but not all proteins are enzymes, because you have many kinds of proteins. Liliane, in terms of your question, or in terms of your answer to Neo, if you're referring to replication, it's actually helicase that opens up the DNA, not DNA polymerase. My understanding is that in transcription, the RNA polymerase is able to open the DNA all on its own without having helicase help. And I don't know why that is. It could be that you have to open DNA a lot more for replication than you do for transcription, or maybe because you're copying both sides. I'm not sure why that is exactly, but helicase is only active, as far as I know, for replication, not for transcription. Bhavna, no, that one came up before. No Punnett square problems. Okay, Shannon, DNA polymerase is the enzyme that matches the complementary bases during DNA replication. Good description, yes it is. How could you know that? Because the name is DNA polymerase and it makes DNA polymers. Again, you guys, can't remind you enough. Use word roots, use logic, use whatever you can if you're not sure of the answer to a specific question. Mira's asking about the Human Genome Project. Was there an ethical issue with that project specifically, or just with the idea of sequencing DNA in general? I think with the idea of sequencing DNA in general, it just made it clear to people that we were going to be able to find out all this information, right? So it was more around the idea of having that information. So a lot of issues having to do with privacy, with how that information might be used for testing. I don't know that anybody was too worked up about like the original project in the sense of like, you know, let's sequence Craig Ventnor's DNA, right? Who's one of the guys who worked on the private genome project, or James Watson, right? I don't know if people had necessarily had an issue with that, but they knew early on, like before they even started, that just doing the project was going to bring up all kinds of ethical issues. So part of the Human Genome Project, you know, part of the money to do that, part of the work of doing that was literally set aside to study the ethical issues and to think about them and think about what we needed to do to deal with them. So that's considered part of the Human Genome Project. And literally, if you look it up, you'll find it's not just the data, but it was all the work that went into thinking about the ethical components of doing that work. Aditi's asking, why is there extra genetic material in an organism's genome, like the introns in mRNA? Sometimes it just gives us flexibility in terms of having more variation. Sometimes it's junk left over from the past, like maybe a gene that's not used anymore and has become defunct. And in the icefish video, you saw how genes can kind of become not used over time and kind of turn into so much genetic junk that doesn't do anything anymore. Sometimes we think it's extra and later we figure out that it has a function. That happens. Some of it is there because it got put there from horizontal gene transfer, right? Like maybe it got inserted from another organism at some point. It never really was useful in us. But it's there. Sometimes it becomes useful at some time in the future. You know, DNA is a living, I don't want to say it's living because it can't really live on its own, but it's evolving, right? I mean, the story of organisms evolving is you know, largely the story of our DNA evolving. So, you know, there's going to be parts that are being actively used at any given time and other parts that are not so actively used, parts that are changing, parts that stayed the same for eons and eons, right? So it's an active thing. But so there's a lot of reasons that you could have extra genetic material. Sometimes genes duplicate, actually fairly often, right? So sometimes genes make copies of themselves for a variety of reasons, and that might generate useful variation to us in the future. Might just create extra stuff. Julie's asking about something on the proteins PowerPoint. It says that protein receptors receive messages. Are those messages hormones? Yes, and other things as well. Right? So we talked mostly about this idea of hormone messages, right, where the hormones would bind to the receptor. Often those hormones were proteins, in which case they would most commonly bind to a protein receptor. Talked a little bit about lipid hormones that could go right through the cell membrane. Not your question. But are hormones the only kinds of messengers? No. You have a whole bunch of messenger kinds of molecules. Cytokines is the word that comes to my mind. So your immune system has a whole bunch of messages that it can send. Where immune cells will send out proteins that kind of alert other cells to do things. That's very common in your immune system. You have other kinds of messages that are proteins and other kinds of molecules, but proteins are often messages because proteins are good at binding to other cells or other proteins and making them do things, right? So they make good messenger material. So I would say if we're talking about a receptor on the outside of a cell, nah, you could have receptors for other things. You could have receptors that bind to, you know, antigens are in a way kind of receptors, right? In the sense that antibodies are binding to them. Sort of, kind of. I don't know. That word might have a more technical definition, but this idea of communication doesn't have to be just hormones. Roghov's asking about the names and functions of enzymes used during DNA replication, transcription, and translation. Just the basics. So you could get all crazy, but we didn't even do that in this class. But should you be able to comfortably use terms like DNA polymerase and RNA polymerase? Yeah. So the stuff we hit a lot, but not all the... There's a bunch of minor little enzymes that we didn't get into. Hey, Eric, haven't seen you so far here. The different disorders from non-disjunction. Yeah, that came up earlier, and I said no, not specifically. Colin's asking about extra credit from the big practical. That's going to go on at the very end. Just because of the way School Loop works, I don't need to get into the details of it, but it kind of makes your progress reports look funny if I put it on earlier. So I just group it together and do it at the very end. And the way it works in School Loop, you might remember from the first semester, is that it's actually a separate category. So there's a separate extra credit category that's counted on top of the normal 100%. And then it's set to a certain amount, like 1%, and then the points come out of that. Okay? So you'll see that appear probably later this coming week. Megan is asking about the rungs of the DNA ladder. So we have our backbones of the DNA ladder, right? And the way that you want to think about that is that the backbones, the parts that are on the side of the DNA molecule, they're really just for structure, kind of make it strong, right? So that's the sugar and phosphate. They're the same for every single nucleotide in your 3 billion nucleotides that are in your genome, right? Sugar, phosphate, the same sugar, the same phosphate, doesn't matter. The rungs is where all the information is, okay? So that's where you have the sequence of the nitrogen bases that stick into the middle that we call the rungs, okay? So if you think of a book, right, the outsides of the book, the paper that holds it together is like the backbone. It doesn't change the content of the book. The content of the book is the words inside, right? So if you think of it that way, those rungs are the information that you would literally read down, the sequence, right? The backbones are the sugar and phosphate that are really just structural. When during the summer do we get our score back? During the summer now. During the summer, we're all taking a break. I think you mean when am I going to post the grades? I always endeavor to have those posted by the end of that week. So they should be posted by Friday, this Friday at the latest. Then we're done. Take off. Have a good summer. Eric, will size and scale be important on the final? It's referred to on 1 or 2 questions, but I would say it's the concept that's more important. Like, by now you guys should have a much, I hope, as challenging as size and scale seemed to you at the beginning, I hope when you reflect back now that it doesn't seem quite as challenging, that you have a better idea of these molecules in context, right? That you have a sense that glycogen is way bigger than glucose, right? Because you understand that we store lots and lots of glucose in molecules of glycogen, things like that, right? So in the sense that there will be lots of references to different molecules in the final, but there's not going to be a lot of specific questions about their size and scale. There is one that I can think of, but other than that, just be comfortable with all the different molecules, right? And it's a good way to practice your comfort level with those molecules and cells to sort of think in terms of what goes where, right? But it's not going to be a lot of specific questions about it. Frank, do we need to know case studies like skin color? Well, again, think about that. Think about the fact that our 2 main case studies, actually there were sort of 3 main ones, right? We used in pretty much every unit of the year, right? Because we started talking about lactase and, not so much skin color, but we started talking about lactase and blood sugar really from the first unit, right? Certainly in cell biology, certainly in genetics, and that's when we got to skin color. We even talked a little bit about molecules related to skin color in cell bio, but certainly by genetics, we were using all 3 of those. Evolution, ooh, they all came back. Physiology, they all came back. What a coincidence, right? So yes, I would expect to see references to those case studies and to those 3 examples throughout the final. Not throughout, but multiple times. Yes, some are at the end, and then some are, there's a few things sprinkled throughout. But yes, there are some in the integrative section. Approximately how many questions will be on biotechnology? To my best I can remember, Bhavna, there's 2 or 3 in the genetics section, I believe 2. But there's a few references to biotechnology that are sprinkled in some of the more integrative questions, okay, and maybe a little bit in evolution. Oh, thank you, Mira, for contributing to the question about the DNA structure. Okay, now I went through the percentages a bit earlier. Yeah, you got it. That's exactly the percentages. Nice work. Victor, relaxed selection, like the kind that happened with the ice fish. Actually, I think there is one question that mentions the ice fish. I can't remember what it's in reference to now, but the ice fish come up at least once on the final. It's not going to get into a ton of detail about a concept like relaxed selection. It might be referred to in one part of one question, that sort of thing. But, it was kind of like one example of this idea of the evolution of genomes, right? So there might be a reference to that concept, but it's not going to be, something that came up one time in one context on one day. Yeah, you might see it referred to one time on the final, something like that. I try to make the concepts relatively similar to the amount of time that we spent on them. If we spent a lot of time on things, they're going to probably come up multiple times because they're integral to understanding biology. Things that came up fewer times in class will show up fewer times on the final, in general. Sham is also asking about extra credit stuff. Yeah, that is your extra credit. That's the only extra credit we have this semester. So the extra credit that I referred to that's going on at the end of the year, that is for your extra credit for your group performance on the group practicals. But if people did retakes, a few people have asked me that recently. No, that does not go into the extra credit. The extra credit applies for the first round through, right? The retakes were really just to help people who wanted to raise one of their earlier scores. And I have no idea about report card Shum. I'm not in charge of that. My grades should be posted by the end of this week. Let's see. It will be at least a couple of weeks, that would be my guess. No, Sofika's asking about the extra credit on the group practicals. So as I said at the time, it would be whatever the lowest point that everybody in your group got. So if everybody in the group got at least a 3, even if some people got 4s or 5s, then your whole group would get 3 extra credit, right? If one person was not able to identify, then you don't get any extra credit, okay? But if everybody was able to get past identification, or at least as far as identifying, then everybody in your group got some extra credit. Mira, good question. Blood sugar and blood glucose. No, in practice they mean the same thing. Blood glucose is a little more technical. That is the sugar that's in your blood. If there are other sugars in your blood, nobody ever talks about it. There might be, I don't know. But when we talk about blood glucose and blood glucose regulation, or blood sugar and blood sugar regulation, we mean blood glucose. So, and I tend to use both of them. I try to use blood glucose, but I say blood sugar sometimes. Don't stress, Polly, it's okay. Really, if you think back to the first semester final, remember that a lot of the questions were fairly big picture, right? I really tried to think in terms of what do I want you to walk out of this class knowing, okay? Generally speaking, people do fine on the final. It's not, people don't generally do much worse than they've done all year. In fact, some people do markedly better. So try not to stress. Get a good night's sleep, okay? Speaking of which, we'll wrap up pretty soon here. Details of DNA replication. Not vast details, no. Again, there's going to be one question on DNA replication on the final. And it's going to be more big picture in terms of why cells have to do it. It's not going to go into vast detail. We just didn't cover that. Most commonly missed. Some of the ones that tie together, if I remember correctly, some of the ones that tie together, like carbon and biomolecules, you know, people tend to still struggle with biomolecules all year long. So I think that's kind of, for people, can be challenging, right? So just really try to, you could try tracking molecules through your body and track them down to the level of the atoms. Like if we did some questions about, you know, how would a carbon atom get, you know, from the air to a plant to you, to your food, to your parts, right? If you could sort of track that through so that you feel really comfortable with the idea of molecules and where they go and what happens to them, I think you'd find that helpful. How do you grade finals so fast? That's why I use a Scantron, Mira. Yeah, I know. I really tried to turn the grades around fast because we all want to go home and be done, right? So, and I know you guys want to know your grades. So let's see. I run the Scantrons through a machine. It tells me how many you got right, and then all I have to do is enter the numbers, right? And I tried to have everything else in there. The only other thing I have to put in is your last learning logs and reflections, which I will be scoring this next week as well. And so all I need to do is enter those scores and enter the scores for your finals. And then I just do one quick look-see to see where people are and make sure it all looks good. And that's the end of that. So yes, should be done. I have my last final on Thursday morning. So I'll be getting those final grades in hopefully Thursday afternoon. Will there be select all that apply questions? Ashwin, if you mean like the multiple choice, if you mean multiples, like where you have matching, there aren't any matching on the final. It's just too hard to do with a standard Scantron. But there will be multiple choices where it's like Roman numeral 1, 2, 3, 4, and then you have to choose which ones. And there's quite a few of those. I use those fairly regularly on my tests. So there are quite a few of those, but not multiple matching in the sense of like A, B, C, D, E, F. Okay. Victor, can recombinant technology be done with a vector virus or manually with other tools? Traditionally, a vector virus it was the main way, right? That we would insert a gene from one organism into a virus and sort of infect a second organism with that virus, right? Now we're learning easier, better ways. I mean, that's what CRISPR is about, right? Is that you can literally cut and paste a chunk of DNA from one organism to another. So we're gradually, and there are other tools actually besides CRISPR now that we didn't go over in class. So now we're in a time where there are multiple tools for doing recombinant DNA technology. And other organisms can also be used besides viruses, other kinds of vectors. Bacterial plasmids can be used, et cetera, et cetera. Everybody's contributing the idea of the Scantron, yes. And yes, that's all I do. Oof. Does that stand for something here, or is it just oof? It's okay. I know you don't have to think in terms of grading. Don't worry about it. But yes, that's how. You are popular, Mira. It was a good question. Eric's asking about for the day of the final, can we sign yearbooks? That's a great question, Eric. So I will tell you the issue, thanks, Mira, that comes up with that. You know what's going to come up with that, Eric. Lots of people will finish the final early, and there will be a few people who feel very strongly and dedicated about using every moment of that final exam period to re-examine every question. I don't necessarily advise that, but people are entitled to, right? So I do have to be a little bit picky about people in their yearbooks, because if I'm not, what happens is people start passing their yearbooks over somebody who's taking the final, which is not very helpful and highly distracting, right? So I will say no passing around of yearbooks. If you want to quietly read your own yearbook, you could do that. But if it starts moving around the room, I might have to come and get it. Not to be mean, but because I need to protect people's time and distraction-free zone to take their final. So in practice, Eric, probably in any given class, at least a few people will choose to take the entire time. If everybody finishes and there is time left over, then of course we can hang out and sign yearbooks, but I cannot guarantee that at this time. Yeah, and I think OOF applies to that question as well. Okay. Well, if there's nothing urgent, that's actually kind of a good note to end on. Oh, we have another yearbook question. Can we give you our yearbooks before the exam for you to sign? Oh, that's a great idea. And I'm sure the tutors would love to sign them as well. They might want you to sign yours. I don't know how they're handling that, but they'd be happy to sign them, I think. I don't have a whole lot for them to do. They can help me clean up a little bit. So they might be puttering around and cleaning up during the final. Be prepared for that. But yeah, Victor, I'd be happy to sign your yearbook. That would be awesome. I was able to have more time during some of the classes than others last week. So definitely feel free to plop that on my desk towards the beginning of the final exam period, and I'd be happy to do that. Meghna, the separation fork. Wow, you're coming up with some obscure ones today. It looks like this, right? All it is the little part where the DNA opens for replication. Don't worry about it. It's a minor detail, right? In fact, it's actually called the replication fork, I think. It just means the part where the DNA is sort of constantly opening up like that in order to be replicated. Not that important. It's like a fork in the road. Okay. So any last few seconds here, if anything critical. Cloning. Not really. I actually kind of took cloning out this year. So don't worry about it. Histones. Histones are those little proteins that the DNA wraps around. So we used to think they were just structure, right? Just kind of a way to keep the DNA from getting all tangled up. Now we know they're actually a little more important than that. They help with regulating gene expression, right? They're proteins, and they can respond to molecules on them and actually help the DNA to be tighter or looser, to unwind or wind. And if you want to be able to use DNA actively, you need it to be unwound, right? So histones can help with that. If you want to make it so the DNA cannot easily be used, you can wind it up, and the histones can help with that. So some of the transcription factors and epigenetic tags work by attaching to the histones and altering their function in particular areas of the DNA. Aren't they just extra coily things? I like that description. The coil is what we call the shape of the DNA. We say that it coils, right? And what it does is it coils on its own, and then those coils coil, right? And then they coil around histones. So coil just means to sort of loop around. The transcription factors, Rivik, those came up a little bit earlier as well. Those are typically proteins that activate transcription, right? Because it's not really RNA polymerase that activates transcription. They're just little machines that do transcription. They're all floating around the nucleus, right? So for them to know where to land and what part to transcribe, that's actually the transcription factors that do that. So they gather in the right place in the DNA where transcription needs to occur. They sort of help the RNA polymerase to bind, and then that's able to make transcription happen in a certain place at a certain time. Last question, Magna. What happens during an infection? Well, again, that's actually one of those broad questions. So infection just means that it implies that pathogens got into your body, right? They could be floating in your blood. They could be infecting specific cells. Partly depends if it's bacterial or viral. That's why your immune system has to fight an infection on multiple fronts, right? It's got to be able to attack pathogens in the blood. That's what antibodies are for. And it might have to kill cells that have been infected. So infected just means a pathogen got in. It could be at different levels. Oh, thank you, Victor, for your description of transcription factors. You're welcome, Meghana. I hope I'm pronouncing your name correctly. One last one. We don't need to memorize the non-disjunction disorders and their chromosome orders, right? Not sure what you mean by chromosome orders, but no, you don't need to know the non-disjunction disorders. Paradigm. Depends what you mean about paradigm, Bhavna. It's a nice word. It's not, we use it in terms of a paradigm shift, right? So this idea of making us think completely differently about something. So that's come up in class a few times in terms of really important changes in science. So, for example, understanding that epigenetics is considered a paradigm shift. CRISPR might be considered a paradigm shift, right? These major things that change the way that we think, right? We say there's a whole new paradigm to think about. I'm hearing a comment from the peanut gallery, but I'm not sure what it is. Promoters start transcription and codon starts translation. Yes, that's true, Rivik. Do promoters bind to the start codon? No, the promoter is in the DNA for transcription. A ribosome binds to the start codon. Oh, you're welcome, Aditi. I'm glad it was helpful. Yeah, I think you're going to do fine. I'm not too worried. Whew, I'm super excited for summer, Eric. Starts on Friday, as soon as grades are in. And my office is packed, and I leave. I'm super excited. I hope you are too. I hope you all have some great plans for summer and you're going to enjoy yourselves. Lots. Don't work too hard. We work enough. That's what summer's for. You won't need to do translation on the final, Raghu. You'll need to be able to describe it. You will do a little bit of transcription, however, or replication. I can't remember. One of those two. You're most welcome, Evelyn. Yee? Oh, you're welcome, Julie. I enjoy it. I'm glad it's helpful. Thank you guys for bearing with me this year as I've tried different kinds of new technology. It's been fun. Lymph nodes. Lymph nodes is part of your lymphatic system, so you have a lot of lymph organs like your appendix and your spleen. So the lymph nodes are just like a little place where your lymph can kind of collect and the cells can multiply, and the idea is they're distributed around your body so that they can sort of more immediately respond to different parts of your body. So you have a lot of them like at the junctures, like where your arms come in, you have lymph nodes in your armpits and in your groin where your legs come in, and I think that just allows a place for all the lymph vessels to come together, and then everything is a little bit concentrated in those areas, and they can respond, and there's cells that kind of filter the lymph as it comes in, for example, from the arms and legs. So it probably helps to filter things. For example, maybe you had a cut on your arm or leg, and it could filter out some pathogens at that point, right? So it's just part of the function of your whole lymphatic immune system. You are most welcome. Whew. Yes, indeed. Awesome. Yeah, I need to have a little open-ended, I should have done that on my last survey for you guys. I'd love to hear a little bit about what you're doing for summer. We'll have to figure out a way to incorporate that so I can just get a little quick feedback. I hope you're all doing something fun, though. So you all get a good night's sleep and relax. It's not going to be so bad. It'll all be over. You guys have worked hard. You know so much. I'm so impressed. I'm so proud of all of you. Nice work this year. If you mean the live streams, yes, I plan to continue to do those. I'll have to work on some other cool technology stuff. If you guys ever hear of anything, you can always pass it on to me. So take care of yourselves, be proud, be happy, and I will see you one more time for final exams. Yay! Yay, and sad too. I'll miss you. Take care. Okay, I'm going to wrap up. Good night, everybody. My last live stream of the 2017-2018 school year. Wahoo! Good night, all. Oof. Happy almost summer. Good night.